And welcome to week 10 of the course where we cover module 10 of this course about RNA-seq methods and the transcriptome. I would like to say that this lecture is going to be kind of two parts. Again, something where we cover the technology and concepts, so more of the conceptual background. And the other part is going to be more on data analysis. And you will see in this module that hopefully you will get a little bit of different flavor where we dive into the downstream analysis a little bit more as compared to the previous modules. In general, we have been covering many concepts as well as trying some tools, looking into alignments, assembly, and these different concepts. So some of the things that we will go through in this module will seem kind of a revision, which is fine because we're aligning this now under what's called RNA-seq methods. First of all, I hope that you are progressing with your own project, as well as different tasks in this course, and I hope you're doing very well in other courses as well. A couple of comments on the last assignment. So my good advice always for anyone is to start early with assignments. I've stated this earlier. This course is special in terms of arrangement and coordination between a group of instructors, which means that some of the assignments, there is a high chance that some assignments would have been prepared by another instructor, which was the case for the last one. And so I reviewed the, so I've looked to the assignment and things seemed, just fine. Until, of course, some of you realized that there were some version issues. Now, although this ended up to be frustrating for some students, some did overcome this by looking to the documentation, try to fix the issues. There were two paths to solve this issue. One was trying to figure out whatever was the previous version, try to install it, and this get done with the existing snippet of code. Or try to look at the documentation and fix some of the parts. Now, the point I'm bringing this up again is to let you know that this is very common in bioinformatics. This is very common, meaning that you may end up running some tool. And in fact, I'm happy that this happened. Like if this was designed purposely, would be really great. Because when I looked at the assignment, it was very straightforward. So it's nice that you have some challenge of that sort. In real life, we face this a lot. And you cannot complain and say, my instructor did not give me the clear instructions. You will end up having a tool from one of the developers or users, and you still feel the urgent need to run that specific tool. So what do? You either figure out the right version, you end up working with them in different machine, you end up trying to look into the documentation, communicating with others. In any case, I made sure to offer to extend the deadline and offer, then we went through the issue in detail. I want to thank Christopher for sharing some hints on updating the code, and also worked with the lab or my TA to offer more one-to-one lab sessions. These are not easy, and by the end of the day, the TA is also a student. But again, we are trying our best to give the support needed here. Though I would like to emphasize, this is a master's course, so more of a self-learning path and process is needed. And I received this point, which is, this is coding. We're not expecting we need that much of coding. And sincerely, this is not a lot of coding, okay? A lot of coding, I know it from my computer science background. I know what does it mean. It means, tens of lines of code for one specific assignment. not 4 or 5 lines where you end up copying most of them and then changing maybe a couple of lines. Anyway, this doesn't mean that it's something to underestimate as a skill, or to say, it should be straightforward for everyone. I would realize that there would be some issues. Okay, so what I'm hoping for is take advantage of communicating with me directly, coming to my office hour or the TA office hours. These should be very valuable resources. Other than this, I want to emphasize that based on some team meetings, some seemed, or discussions, some seemed will progress with their project. At this stage, you don't want to be in a case where, or in a shape where I don't know which results I'm going to produce or reproduce. Okay, and I gave the general advice for everyone, and I've said this in the lectures before: try to simplify your task, aim for a specific part of that study, start with it, or go to the code, shared code, and see what it gives you, and then start to get to line this up with any of the results section. Simplify your task before you take it to the next stage. And then another common question: is it enough to run one thing? It depends. If running one thing was pretty complicated and you're going to report your experience, share it with us. You are going to present this as a team, where you will come as a team and try to present this. So if we're going to feel that this is sufficient, or altogether you're going to report your experiment and your experience as well. So maybe one experiment or one result is pretty sophisticated to reproduce as compared to very simple, and then you take it to the next level. All of that will be considered in grading. In this lecture, there will be a set of discussions about RNA-seq. It is focused on RNA-seq. RNA-seq does not mean one thing or one type of output, as we will see in the coming slides. But in generally, by now, as experts in these concepts, we are looking at taking the RNA, fragmenting the RNA, and looking at the reverse transcript. You get the cDNA, and from there you apply the next generation sequencing. Then you are able to measure certain outcomes. We'll look into this. Eventually, we have to deal with certain challenges. And once we are done, we go through the machines, we get some output. We want to analyze that output. So I want to show you and make sure, hopefully in this class, you get to see a little bit more about data massaging, data mining, data science, machine learning. In this lecture, without diving so much into these definitions, these terms and what they mean, because sometimes they are big umbrellas, we're going to touch on some of the thoughts from all of those. Something common in between is what we refer to as multivariate statistics or statistics. So of course, we'll have to visit some statistics and see some visualizations. At the end of this lecture, there is an in-class activity that I have designed for this lecture. It's going to introduce you to R programming language. So what I want you to do is ease yourself a little bit, open that exercise online, look into the resource that I've shared, read through, download the R code. R is very common as a programming language in bioinformatics. Python and R. When it comes to bioinformatics, everyone knows R is dominant, as well as Python, but I would say R first. Okay, given the load of tools are there and written in bioinformatics. So what I want you to do is try to make that exercise. You need to do a minor change. You have all the instructions. And then the assignment for this week as well that I have designed is going to take you through several modules in this course. This assignment is going to take you through dealing with the database. I've already prepared the raw data files from the gene expression omnibus. So I made these available because you will end up spending lots of time downloading the files and then putting them on server. And also I had to reduce these to make the next steps more smooth. However, that's it. So you have a set of raw data files. What you need to do is everything else. Downloading the reference file, doing the alignment, getting the results, making some post-processing, cleaning the file, going online on one of the tools, taking the steps through normalization, differential expression analysis, enrichment analysis, and sharing some visualizations, and sharing all of that as a type of report. So I'm pretty excited for this section of the course, and several things were designed to combine several, different things that we have been doing. Now, some of you might say, okay, finally we get to the data part in this course. Let me remind you, this is not the data mining or data science course. These you would find separate courses for. Personally, I would have designed the whole course about data analysis, data science, because this is my own area of expertise. But this is a bioinformatics course. You have to be acquainted with genomics concepts, cell biology concepts, and so on and so forth. You have to be acquainted with skills into running different tools. Running tools for the last few modules were core part of knowing databases and so on and so forth. So I've already covered a little bit of what may relate to learning objectives. We'll look into the RNA-seq workflow and some different methods. Last lecture, we looked into ChIP-seq, for example. We go quickly over that again. There's RNA-seq and single-cell RNA-seq. Also interesting things to consider when it comes to RNA-seq. What is a transcriptome? What is a genome? By now, we probably understand, we are looking into splice junction areas and how we may end up with exomes only, for example, as compared to a complete genome. What kind of steps we need to follow when we are analyzing, in general, these types of data sets like RNA-seq data? We're not covering microarray data here, but pretty much the workflow is, in terms of steps in general, would be the same. We're always going to start with normalization, filtering, and we keep going with other steps. Yet, of course, there will be different unique tools depending on the specific type of data that we're dealing with. So here we're focusing on RNA-seq. Why we are learning this? Because as a bioinformatician, and as I play my role in different projects, a valuable output from me as a bioinformatician is to inform the decision-making process, whether this is a pharmaceutical company or it's a government institute and so on and so forth. So to be able to deliver on that side, it means I need to figure out the right steps to execute certain type of analysis. I need to produce a set of figures. I need to communicate through analysis, and everything would line up. So if we mess up treating the raw data, if we don't do the proper normalization, the quality check, and so on and so forth, and we don't report this sufficiently, we are not actually informing the decision-making process, which eventually could really impact the health, the environment, and many other applications. So in this first part, we are going to talk about RNA-seq. RNA-seq analysis. When we say RNA-seq, it means we're looking at the RNA molecules. So we start with some samples of interest that we want to work with, and then we're going to isolate the RNA. And based on, of course, once we are isolating these RNAs from the DNA sample, we are able to categorize, or maybe we are able to pinpoint the areas of interest, like coding areas through poly A tail, for example. And then we get the complementary cDNA, that's the complementary DNA. So then we move to the sequencing part. Pretty much we have discussed this through the different modules and why we need to do this and so on and so forth. We end up with sequencing both ends. If we're doing pair ends, that is more accuracy. So then we move on into matching the RNA reads. Eventually we can move to some downstream analysis. So the key terms or concepts of RNA is, of course, going to be RNA isolation, reverse transcript, that is complementary DNA, and then you move on. Now, whether you do this in a targeted manner, meaning I'm going to focus on a set of genes, that is targeted RNA-seq. whether I'm going to do this based only in poly A tail, that is poly A RNA-seq, whether I'm going to, look into mRNAs, and whether I'm going to do this in a single cell fashion, focus on a cell specifically, that is single cell. So we have all of these variants that we can dive into as part of our RNA-seq methods. So mRNA sequencing, for example, is one of these methods. It's similar to poly A RNA-seq because in poly RNA, the method selectively sequences mRNA molecules by targeting the poly A tail, which we know that's found in general at the end of most eukaryotic mRNA. So similar to that type, this method, which is mRNA sequencing, focuses on sequencing mRNA molecules. And eventually what we get from this is gene expression. That is starting with the samples, looking at the total RNA sequence, okay, and then doing the fragmentation, sequencing all of these, aligning as per specific genome or transcriptome, and then we're able to know what is the quantification of one specific gene, let's call it gene X, in sample 1, and sample 2, and maybe those in sample 3, and maybe these are control samples. Maybe have some treatment group, like sample 1, T, sample 2, T, sample 3, T. And so I want to know the score of each of those, okay? And then make a comparison as a group to group, okay? See, do we have in general different distributions? Do I have this shape of distribution here and another shape of distribution here, or both they look the same? Okay, so maybe if I sketch this again, but because the core of what we are doing with the mRNA, with an mRNA RNA-seq type of analysis. Just bear with me a second. Okay, so here, what we want to do is we have our gene of interest, call it gene A, and then we have maybe gene B and maybe gene C, and so on and so forth. Usually, by the way, these could be transcript IDs, as you will see in the exercise. So it's going to be your own task to map those to the genes using some reference library. So depending on the output that we receive from the machine, we don't usually get the gene names directly or the gene labels. So in this case, I may have some control group. Maybe I have replicates in these control group. And then maybe I would have some treatment samples, okay? So maybe I have maybe exposure 1. This is maybe, let me call it A1, A2, exposure type A, exposure type A, third replica. Okay, and then I have scores, a set of scores for all of these different genes. These, of course, are the transcript counts, sort of, right? We're doing RNA-seq analysis. So we want to know, again, what is the quantification for all of those? This is the output of mRNA sequencing. And so we get this output here. We can end up with one more or many more types of exposure. So we can have exposure type B, which could be, this is just a high dose. This is a middle dose, depend on your own design. This is exposure B3, and then we have also numbers. These squares are referring again to some numeric values, okay? Whether those are discrete or real values, it depends on the type of technology and the pre-processing that we are doing. So now we want to see among these groups of different types of experiments, we want to zoom in at the level of one gene and do some statistical analysis In particular, I want to see the distribution here. So I may end up with different distribution. Who knows? Okay. I want to compare this distribution to the distribution of other genes. Okay. Or the same gene, or the same gene at across different experiments. And we're going to see what do we realize for maybe the control distribution. What do we realize for the first type of exposure, third type of exposure? And what is the shift here? It's not easy when I draw it this way, okay? Because when I put them on top of each other, this is when we start to see things. So in other words, I may end up having something of, oops, if I move this to here, maybe depending on the statistics that we do, okay, I'm going to end up with this coming over here, okay? And maybe the third distribution, which is this one, I want to align it here, is going to be in general very close to the second one. So then I want to see, and again, when I say distribution, I mean I'm going to consider the values across these experiments and see the pattern that they follow, and then maybe model this with some function and see overall how much they align on top of each other. Maybe that's helpful to say, oh, they are unique. There is an area of tolerance that I accept to say these two groups are different. So this is basically the downstream analysis as we start with this, as we start right with the mRNA-seq and so on and so forth. Okay. Oops. Yep. Another type of sequencing is single-cell sequencing. Single-cell sequencing is an advanced technique. I would say it's very interesting that we have now single-cell RNA-seq sequencing because it brings our attention and focus to gene expression profiles of individual cells. So now, as compared to the previous drawing that I had shown you, instead of looking at gene A, of course, I'm going to have gene B. Now, instead of looking at control experiment 1, I'm looking at cell. Okay, of course, these could be different experiments. We can label them, but I'm looking at the granularity of cell 1, cell 2, cell 3. This is really amazing. So I'm able to measure now gene expression profiles for different types of cells. This is, of course, I would say, advanced and achieves lots of advantages that we can do, especially working with cellular heterogeneity, when we have tissues or population of cells, each referring to different types. Of course, the technology starts with what's called cell isolation because we need a way to be able to isolate these cells. So one of the reported techniques is what is called fluorescence activated cell sorting, for example, or FACS. That is going to, through some laser techniques and light techniques, going to able to capture the information from one single cell. Eventually, we get this data, and doing some analysis, we try to cluster different types of cells. It could be the case that when we're working with, for example, brain cells, we don't know exactly which cells are those, okay? Okay, or let me fix this actually. I'm trying to remind myself of some data set I've worked with for a good time. So I guess we know which type of cells as part of the general. We know, for example, the nervous system and the brain system. We have different types of nerves. And so we have access to that information when the data comes. So I know cell 1 and cell 2, if they go to the same type of cell or group. Now my task is to confirm this by clustering my data and in this type of domain to see if indeed we have this group of cells coming together or not. and so on and so forth. Eventually, I can do some visualizations and look into my read counts where I have gene versus cell 1, cell 2, and other types of cells. Okay, of course, reverse transcription is part of the process. Once cells are isolated, each cell is lysed to release its RNA content. Okay, so there is this process to push the RNA content from the cell. And again, we keep going with what? Reverse transcription, cDNA, amplification, library process, very similar steps to what we went through before. One might ask again, why do we need to do single cell RNA-seq? This is in general, one simple answer is because the cell population itself is heterogeneous. And as you see in these pictures, if we are looking at the human blood, for example, there are different cell types. Cell types. Okay, we're looking, for example, in human blood at neutrophils, some type of white blood cells, monocytes, some other type of white blood cells, platelets, and so these are different groups of cells in the blood itself. I'm not talking about, different source. The blood, I take a blood sample and I have within the blood different groups of cells. I take a tissue from the brain, I have different types of cells, and so on and so forth. So when I look at these different types of cells, like in this case, we have the platelet, and platelets are known to repair vessels and stop bleeding, for example, as compared to neutrophils and monocytes that are known to as white blood cells that are going to fight the pathogens. So what about, again, being able to dive into this and see the gene expression across these different types of cells? This is one type. This is another type, and so on and so forth. This is the third type. And be able to zoom in here. Maybe we have something interesting to learn about. The conventional RNA-seq way, it provides only average gene expression across all cell populations. That's why we call it bulk RNA-seq. As compared now to this single cell technology, we can do it at the level of one specific cell or at this granularity of single cell. Okay, and this is now possible through this technology that is called a droplet-based sequencing. And this is a microfluidics technology used in genomics to be able, again, to isolate these cells. We need some steps. There are going to be some steps, of course, to try to work through and be able to screen or find the information of single or specific cells. So this starts with what's called cell suspension, cells from suspension, and cells are suspended in a buffer and individually encapsulated into droplets. Each droplet acts as an isolated reaction chamber. And then we have microparticles that are coming from here. And again, you can see this illustration here. They'll be joined together, okay? Microfluidics devices create the droplets by combining a stream of what? The cell suspension with a stream of oil. And the oil and water do not mix. This allows for the formation of discrete droplets. So using this whole idea and design, we're able to get this kind of a droplet that will hold what? The information of a single cell. Now, also into this process, along with the cells, okay, along with these cells, the droplets contains beads that are coated with unique DNA barcodes, and You may refer to it, right? The first modules we talked about oligo DT sequences that can capture mRNA from the cells. And again, this would continue through a group of steps, cell lysis, to try to get the push the RNA out, release the mRNA, and then do the reverse transcription, pooling, amplification, and so on and so forth, reverse transcription, until we are able to get the sequencing information reflecting these types of cells. Let me run for you this interesting video looking at the droplet barcoding for single cell.
Hi, I'm Steve McCarroll. And I'm Evan McCosco, a postdoctoral fellow in...
By the way, those two guys are the guys behind the droplet barcoding for single cell. Those are the two guys that led this discovery and pushed the single cell sequencing.
And I'm Oni Basu, a postdoc with Aviv Regev and the KCU at Broad, and with David Weitz at Harvard SEAS.
3 years ago, we started working on a new genomics technology to make single-cell gene expression profiling fast and straightforward.
Complex tissues, like the brain, contain many different populations of functionally specialized cells. These cells use the same genome in different ways. There is so much to learn about the ways in which specific cell types assume unique cell shapes, accomplish feats of physiology, and respond to mutations and other stimuli.
What is the range of cell types out there? We wanted to develop a method to systematically characterize brain cell diversity. If we could get enough gene expression profiles from these cells, we thought we might start getting answers.
When we started this project, gene expression analysis was really good at measuring gene levels in a single homogenized sample.
So it's really like a fruit smoothie, which has all the different fruits commingled and homogenized, which is still pretty delicious. But what we really wanted was the fruit salad, where you could taste every fruit one at a time. What this technique does is measure the genome-wide expression cell by cell.
We started thinking about trying to make a system that would let us make thousands and thousands of single-cell RNA-seq libraries in parallel, inexpensive, easy experiments. We had been using droplets to study many things in molecular biology, such as the expression and copy number variation of individual genes. Droplets provide a way of scaling molecular reactions so that one can perform hundreds of thousands of them in a tiny microcentrifuge tube.
What the microfluidic technology brings to the table is really the scalability. We can do around 10,000 to 20,000 cells at every go, and we can really look at complex systems now, like the brain or the immune system.
The challenge with droplets is they don't stay in one place, and they don't have stable addresses. We needed a way to put a DNA barcode onto the RNA from each cell, and to have that barcode be different than the barcode that was applied to the RNA of each other cell. Evan came up with a cool way of doing this.
We use beads to deliver DNA barcodes into droplets. We synthesize the oligos directly on the beads. To make the barcodes, we split the beads at random into groups, add a different DNA base to each group, and then pool the beads again. Repeating this split and pool process 12 times, we produce 16 million different barcoded beads.
Oni designed microfluidic devices that could deliver these beads and cells together into droplets. We call this technology DropSeq.
But how can we really know if we're making single-cell libraries?
It could have been we were just making a lot of really tiny smoothies.
So we came up with a simple test. Let's put human and mouse cells together in our system. If each bead or cell barcode associates with a mixture of human and mouse transcripts, we've ended up with smoothies. But if each bead has captured transcripts from just one species, then we preserve the information about individual cells. In the first series of experiments we did, many of the cell barcodes we identified had mixtures of human and mouse transcripts. We continued improving the design of our lysis buffer, we increased the stability of the droplets we made, and adjusted the concentration of cells and beads used in our experiments. Eventually, we were able to achieve very high organism specificity for the great majority of the cell barcodes we sequenced, proving that our libraries came from single cells.
So we had DropSeq in hand, and Now we could finally do what we wanted to do, which was to use it to study complex tissue like the nervous system. We chose to start with the mammalian retina because so much is known about the different cell types that exist in the retina, their functions, and there are molecular markers for many of them.
We generated 44,808 single-cell profiles for mouse retina.
It took 4 days to do with Dropseq. Rahul Satija developed computational approaches for clustering these 45,000 single-cell transcriptional profiles into clusters or groups. We identified 39 transcriptionally distinct populations of cells in the retina.
We found cell populations that didn't have markers before. And most reassuringly, we found a lot of types that we did know about.
A single scientist can come in and make 10,000 single-cell libraries every day. And the reagent costs are small, on the order of 6.5 cents per cell. There is so much exciting work to do, and we hope that scientists enjoy using Dropseq to do that work.
This is very interesting. First thing, it gives me again back, should I fix what I fixed earlier, what we get with RNA-seq data. So we get the cell information, and then I think through the analysis we would be able to group cells and profile them together, and then maybe we can tell which types of cells are those. So anyway, I think some data sets are having this data, but I don't know if that's already generated computationally. I would have to check. But I still remember these types of data that we will end up to always with RNA-seq data. We need to do some clustering to cluster, which is a computational techniques. And I'm gonna highlight this method in the downstream analysis that we'll do, which will be, of course, useful. But what is more interesting, of course, in this whole talk, personally for me, is as science and scientists, you see when they started, they made all of their efforts with some assumptions, they tackled the issue, and then the results they got did not show full distinction of the mice or the mouse and the human sample. Did they stop? Did they say we failed? No, they kept trying and refining until they got perfectly what they're looking for. So this is a message for everyone. You are not, and generally in real life things and whatever we are doing, we don't get things unless we are very lucky right from the first time. So we end up trying, and with patience we get what we want to do. So what are some of the differences between mRNA-seq and single-cell sequencing? I put this table for your own reference so you can go through them. Of course, we know here we're looking at gene expression across bulk samples as compared to looking to at the single cell level. So here, this is the resolution in single cell. In terms of application, we are looking into cellular heterogeneity, what kinds of cells we have there, and so on and so forth. This granularity can be useful for lots of applications. Here, it's more of a global gene expression analysis. So what I'm interested with in this type of mRNA-seq is to try to see if this gene is significantly different than the other one. While here, I'm trying to shift my focus on the nature of cells and combined with the genes information. Cost, typically this is less expensive for the time being. This is more expensive. Who knows what's going to happen in the coming days, and so on and so forth. In terms of data analysis, we usually start with differential expression analysis. Here, we usually start with the cluster analysis. The samples are not clear from which cells they are coming from. We know that they are labeled somehow differently, but we end up with a data set showing us counts for genes or cells versus genes. So we need to do cluster analysis and then do some dimensionality reduction and finally try to identify these different types of cells. Another types of RNA sequencing that we have also looked into in a previous module, specifically module 5 here, is what's called targeted RNA-seq. So instead of, looking at all the coding regions, what about focusing on specific genes? And this could save us some cost and in terms of money and time. So here, the core of this is using streptodividine beads and then elution from these beads. And we know that there are biotinylated probes that will be used. Okay, these probes will be attached here, and then these are designed to hybridize with the RNA sequencing. And then streptodividine has a high affinity for biotin. So here it's this streptodividine is added, which is coated with magnetic. It's a coated magnetic P beads. Okay, and then if we are applying some source of or process of magnetic separation, we're able to choose specifically the sequences related to these genes. So right from the beginning, we define some sequence and attach to that sequence specifically these probes and beads, so we are able to separate them. This is targeted RNA sequencing. Another type of sequencing, which is very common for identification and classification of bacteria and archaea. This is 16S ribosomal RNA gene sequencing, which is, we know from previous models that 16S ribosomal RNA is a component of the 30S subunit of prokaryotic ribosomes. In other words, if we're interested in bacteria and these species, this is the way to go. And generally, these species are coming in populations. Sometimes they are mixed, like in the human gut. We have many of these species. So we're dealing with the metagenomic kind of samples. So we end up going through some protocol where we we are trying to define different regions that we can deal with. Here we have V1, 2, oh sorry, this is V1, V2, V3. So these are kind of regions that provide species-specific signature, which are part of the 16S RNA gene. And this is, again, useful for knowing that this is different bacteria or not. And then we do the amplification steps, sequencing, data analysis, and so on and so forth. What we end up usually with after the analysis and the workflow is what's called OTU tables. OTU, or Operational Taxonomic Unit Table, is the common way to represent the counts or abundance of the different organisms. Usually we're talking about, again, microbiome analysis, microbial, and so on and so forth, across a set of samples that we're interested to do. In this table, we would have rows. This is the OTU table, OTU, Operational Taxonomic Unit Table. Rows would represent a different, each row represents a different OTU. So this is one OTU. We're not talking about a gene. Let's say OTUX, for example. This means this is a group of related sequences that we can consider them to be the same operational taxonomic unit. And this is usually defined by clustering sequences at certain similarity threshold as input to the program. Now, columns would be, again, different samples. So this is sample 1, sample 2, and then until we have sample n, for example. Okay. Now, of course, we want to run specific tools, usually Chyme, BioCrust, LibC. These are different very common tools in 16S sequencing. Quantitative Insights into Microbial Ecology, or Chyme, is an open source pipeline for performing microbiome analysis from raw DNA sequences. It supports many steps, and eventually it helps me in or us in doing the taxonomy assignment and the diversity analysis. PyCrust is another tool helpful in predicting the composition of the metagenomic or functional profiling. And LIFC is for high-dimensional biomarker discovery and explanation. Okay. Now, if you're interested to look into, strategies for examining the human microbiome, we can go with different technologies and different steps. We can pursue 16S rRNA gene profiling, where eventually we can do some clustering of these sequences, so we are able to identify, hopefully, eventually, some different groups of taxonomies, some different groups of microbes. This is when we are dealing with, the taxonomies or taxonomic identity for each ETU group of sequences. And the matches, one would ask how we are able to recognize, that it's this bacteria or this species. Usually this is going to be sequenced or looking at the sequence homology against known 16S RNA gene database. So always remember this thing, having a documented way of information, which is the database, is very helpful in many of the things, and many of the tools are using a public database, their own database, asking for an input database from you as a user. So there are always these different ways to look into the analysis. In the metagenomic analysis, in this case here, the DNA sequences can either be mapped to reference genome or through de novo genomes, which we're going to look into in the coming slides. Then the relative abundance of the present genes and the functional potential of these sequences can be assessed. As compared to more, we're pursuing this analysis of the human microbiome. We're going to dive in and find all of the information that we can go through. What about viromics? Okay, the viral particles. These viral particles must be enriched first, and then we can obtain what? Virus or the viral genomes. Finally, we can follow what's called meta-transcriptome as compared to meta genome. Metagenome is looking at the DNA aspect. We have a mix of samples of DNA sequences. Here we're looking at the RNA sequencing, RNA level. ChIP-seq, something I covered in the last module. So quickly going through this, we know that here we are interested in the gene regulation, right? Like looking for motifs, transcription factors, binding sites, and the secrets for those positions of these seeded messages is to look at the protein-DNA interaction in the genome, the protein-DNA interaction in the genome. And there are this series of steps that we can go through eventually to look into identifying these DNA regions that are bound by specific proteins. And so we go through the chromatin immunoprecipitation sequencing, which is the description for the ChIP-seq acronym. Now, usually the workflow process involves cross-linking DNA and protein, fragmenting the DNA, immunoprecipitating the DNA, and so on and so forth. And again, different applications are there for that towards studying gene regulation, epigenetic modifications, and so on and so forth. Nice. So now we have looked into an overview of different RNA-seq methods, different applications. Now, next, we're going to start more of looking at RNA-seq alignment. And when we talk about alignment, we understand that we got the reads, and now we want to understand what those reads mean. So the focus here is going to be more on mRNA sequencing, which is the most common approach that we find around with many, many applications. So this technology that is very prominent, accessible in many places. How can we handle that? And what are the different approaches for that? What if specifically we have a genome? What if we don't have a genome? And so on and so forth, which is going to bring me to the one slide here. Okay, so I'm going to come to this. But first, as we establish, let's say, a map of our biological system, we know that there are different layers that we can work with. Looking at the genome, we're looking at the full DNA, whether it's coding genes or non-coding genes and so on and so forth. When we move to the transcriptome, we're looking at the specifically maybe coding mRNAs or non-coding RNA, and then looking at the proteome, we're looking at these interesting structures of the protein. So we have these specific proteomics techniques that will help me to analyze the structural and the function of those proteins from a different aspect. Yes, transcriptome is what led to the proteome, but again, there are things that are very interesting for us to look at these amino acids and the metabolome, right? Metabolome is the other chemicals we have in our body, amino acids, sugars, lipids, all of these small kind of chemicals. And microbiome, and you keep going, okay? Different layers into how we can establish this whole thing. All of these are really big topics. So I would say so far there's very good progress in genome, transcriptome, proteome is there, metabolome is also there. And probably it's one of the very big things. And everything here is really a big thing, okay? And maybe in the future we'll be able to say, maybe those are gonna come all together, because so much what we see, they are generally treated separately. So one lab is doing one experiment with that protocol, another with another way. What about what's called multi-omics or meta-omics? That is still limited given the universe of accessible experiments for each. In other words, you will be able to find several studies where they combined several of those, but again, they are very limited. And even if they are, you would find a set of accessible data sets, they are not as common as for the other types. It's going to be very interesting to establish more and more studies looking at the proteome, genome, metabolome, transcriptome at the same time, and really pinpoint things like specifically for cancer diseases and so on and so forth. So when it comes to read mapping or alignment in general, either I'm having a genome, okay, it's either I have a genome so I can use it as a reference to come with my abundance counts, or I don't have it, so I have to build a de novo way. This is what we refer to as de novo. So which means something we discussed before, we don't have a reference, so reads need to be assembled first into longer context. That's what we call de novo assembly. Genome mapping. This is a very nice slide, and I think this can be a very good reference to bring everything together in terms of alignment. It's either we have genome, okay, we have a well annotated genome, so I can go to ensemble database and I can find the GFF annotation file, which is the annotation file. We'll see one example today in the module, and then go through some process of, read mapping to the genome, and then getting the results after analysis. Finally, I can do functional annotation. As compared to transcriptome mapping, where here we're going to ignore introns, focus on exons, so also depends on accessible transcriptome file. The assignment is about this type of analysis. So you will have to download the transcriptome, and then you can actually go and align the reads using a tool called Callisto and find the counts at the end. What if we don't have neither genome nor transcriptome? We end up doing a de novo assembly. So we need to do the assembly, usually construct what's called De Bruijn graphs. And this is very, computationally intense process. And then we build assembly through solving the puzzle, the jigsaw puzzle, and we also can end up with functional annotation and using some very common tools to be able to recognize. Because one of the things when we don't have a genome is doing annotation. What is this sequence? So through analysis of homology and orthologs, we may be able to derive that this area would probably link to that gene, so to that specific function based on the databases that we have. If I compare transcriptome versus genome alignment, genome means DNA, transcriptome means RNA. And relatively, transcriptome is simpler because introns are out. Okay, yet there is some challenge, okay, because we can deal with alternative splicing, the ordering of exons in the process of transcription. So this can lead to multiple transcript variants. Okay, so here we are looking into finding the expression of specific genes, right? Here we are looking to find the structure or location of genes and detect as well genomic variants. This is not generally for genomic variants. So if you're interested to know genomic mutations, and so we will go with genome alignment. This is, again, it's a reference table for you to see when we are using different types of tools. Hopefully by now you feel a little bit more comfortable with some of the tools. Nothing like going and trying some of these tools and keeping an eye on the advances. From a few years, they're still dominant in terms of the progress there in the field. So I would say they're still well accepted. What we're dealing with now in terms of new tool development, again, either incremental enhancements of these tools or some speed ups and so on and so forth, or we're looking more at the third generation sequencing like, Oxford nanopores and so on and so forth. Different applications and outputs. So now we move to the RNA-seq. What types of files we are dealing with? Okay, so generally we have the FASTA files and the FASTQ files. Okay, we're dealing with the read files and the sequence files, as well as we are dealing with alignment files. We have talked before. About the different files. So we heard about SAM and BAM. Okay. SAM is again tab delimited file. So BAM file represents the same information, the SAM file. This is the alignment output. Whenever we are doing an alignment tool, we get this as an output. BAM is just the binary version here, so it's more compact for the storage space that we're gonna deal with. Now again, FASTA files that we have here, it contains the sequences in simple format where we have each sequence, and FASTQ is one that we have seen before, okay, which comes with a quality score. Remember when we've discussed the quality score, and this is one of the concepts. So maybe it's an hour and I've not asked a single treasure hunt question. So what did we call that score that we used in the FASTQ? I'm gonna just put the first two alphabets here and ask you what did we call that score that we have discussed for FASTQ files. So we have sequence files, we have alignment files that we deal with. These are the inputs, these are the outputs, but we use, if we have genome file, we use some feature annotation files, specifically generic feature format. These are again tab delimited. We use them to describe the different areas. These are describing for us the reference genomes. There are different versions of those, right? The standard is GFF3 right now. So this is an example. I've worked with those ones. They are, I guess, straightforward. So you have the chromosome, you have the specific source, and then we are told what is the start and end location. So this is where the mapping happens. So we know what is the sequence based on the genome, what sequence is here. And once we are able to map to this, we'll be able to tell if this is a gene or mRNA and so on and so forth. And we may have some attributes indicating more information like the tag information and so on and so forth from this file. So this file is pretty valuable to us to download. That's why if you want to do alignment using genome file, you use the GFF file, okay, because it will help us to annotate and the sequences that we have. So I'm going to stop here and then continue with the next part.